I just want to say thank you for the privilege of coming to talk to you all today. My name's Alex. I'm at Virginia Commonwealth University. And so the microbiome has become, so the microbiome has become a pretty in vogue topic for research in the last 10 years. But it's really most of the research, or a lot of the research, is focusing on humans and understanding how humans relate to their bacterial communities. But as evidenced by the recently announced Microbiome Initiative, woo, there's a growing need, and people are realizing the growing need to start understanding the microbiome outside of humans and to broaden our understanding of the role of bacterial communities. Because one thing that we know about bacterial communities and microbiomes is that these associations are rooted deep in evolutionary time. In both lab experiments and in the wild, what you can see in a lot of species complexes or sets of species is that the microbiome recapitulates host phylogeny through time. The composition of the microbiome recapitulates host phylogeny through time. And this is also evidenced by the fact that microbiome composition is directly related to changes, or directly linked to changes in host health, right? So when hosts lose that kind of species-specific microbiome conformation, or the composition changes, it's directly related to negative physical effects. But one of the problems with the way that we're looking at the microbiome right now, or one of the shortcomings, is that we largely focus on how the microbiome is shaped within the lifetime of a host by contemporary factors, things like diet, things like age and stress, or antibiotic regimens. But a more interesting question, and one that we really haven't begun to understand as well, is how is the microbiome composition shaped over evolutionary timescales? So why does the microbiome appear to recapitulate host phylogeny? So to kind of start at getting at that understanding, our question was, how is microbiome composition shaped by host phylogeography? And we used this species, Odontotaenius disjunctus, or best beetles, to answer that question as a model system. So best beetles, as you can see here, they're little beetles that are about that big, and you can find them under logs, but they're distributed throughout eastern North America, really commonly found. But they're interesting because they're symbiotic, or putatively symbiotic, gut microbiome likely underlies the evolution of two important behaviors. One is sociality, because these best beetles require adults to defecate into the mouth of larvae in order to pass specific microbes, which are thought to confer the second behavior, which is phylophagy, which is wood feeding, which is uncommon among beetles, but it's likely that the gut microbiome is involved in both. So ecologically interesting, but also interesting from a experimental perspective. They make a good model system because we think that largely the microbiome is vertically transmitted, because the microbiome is passed from parents to offspring during development, and also because they eat, they subsist on largely deciduous trees. Diet is naturally controlled for much more than in other systems like humans. So to answer our more specific question here of how has host phylogeography influenced gut microbiome composition, we did this through two specific aims. So first, we characterized host phylogeography and understood the evolutionary processes that explained these extant patterns of genetic variation in the host. And then we determined or explored the impact of that process on the gut microbiome. So we performed a pretty comprehensive sampling across the natural range of the species, which you can see shaded in gray. We took 188 beetles from 23 populations, which amounts to about 9 beetles per population. And to characterize host genetic variation, we used a double digest rad-seq, which is a genome reduction technique used for non-model organisms. And we identified over 163,000 high-quality SNPs. And for the gut microbiome, we characterized the composition using a 16S rRNA PCR survey of gut extracts. And from all individuals, we found in total about 8,000 OTUs, which is basically a bacterial species. It's helpful to think of it that way. So from the host's perspective, one thing that was interesting that immediately jumped out was that host population structure exhibits this classic pattern called the Appalachian-Apalachicola divide, in which you have the dominant axis of population structure here separates populations into larger regional clades. So into the west of the Appalachian Mountains, into the east of the Appalachian Mountains, and then along the divide itself you have the central clade. And so, let me back up real quick. So this pattern is important because a number of species in the eastern U.S. exhibit the same phylogeographic pattern, and usually it's attributed to the species surviving Pleistocene glaciation in two refugia on either side of the Appalachian Mountain, or that discontinuity. And so we have pretty strong support for that in our data. First off, what we see is that the western clade and the eastern clade are reciprocally monophyletic. So that would be what you would expect under a situation in which both of those clades descend from common ancestors or ancestral populations on either side of the Appalachian Mountains. And in the central clade, we have some pretty good evidence for that these things may have arisen from secondary contact. As you can see, this is a structure k equals 2, but western and eastern ancestry are almost entirely assigned to their respective origin, and central ancestry is mostly entirely mixed between the two. So to more rigorously test that, we used approximate Bayesian computation to test 9 different demographic scenarios. And so the point of that is to determine which of these demographic scenarios best explains the present day genetic structure that we observed. And so what we found is overwhelming support for a demographic scenario in which present-day individuals descend from 2 populations that diverged around 20,000 to 30, or around 10,000 to 30,000 years ago, and that those 2 ancestral populations eventually became the present-day east and west clades, and that the central clade arose from secondary contact after that divergence had already taken place. And the timing of this event right here, as estimated, it really implicates Pleistocene glaciation pretty strongly as the mechanism of this demographic history. Because as you can see here, around the last glacial maximum, which is about 20,000 years ago, is right at the center of this 95% confidence interval of the estimated divergence time, which at that time, global temperatures dropped to their lowest levels, and the Laurentide ice sheet right here descended as far down into the continent as it has been. And so that would provide a mechanism or a reason for why those populations would have been isolated more than 20,000 years ago. So then the question is, are host signatures of Pleistocene glaciation reflected in the gut microbiome? So from a presence-absence standpoint, interestingly, not really. Though we found over 8,000 OTUs, or different bacterial species, most of those, over 85% of the total relative abundance of each gut segment was made up of the same 600 bacteria. So these are called core bacteria, and they're unique, and they're found in we detected them in every single individual. So by and large, most of the microbiomes that we found from all of our individuals are the same from a presence-absence standpoint. And more interestingly, most of these core bacteria appear to be new species, or species that are unique to Otis junctus. Actually, over 90% of them have no known analog in existing sequencing databases, which certainly suggests that the bacteria that are in this gut, as has been thought, likely have undergone a long-term co-association with their host here. And with respect to Pleistocene glaciation, this pattern, because they're universally conserved in every host, and because they appear to be transmitted parentally, because they don't come from the environment, or we don't think that they do, it likely means that they were present in a pre-Pleistocene ancestor and have been maintained independently in the eastern and western clades until the present day through parental transmission. But that doesn't explain all the variation. So we then asked what factors explain differences in gut community composition. So we used a redundancy discriminant analysis to look at whether geography, host phylogeography, or the environment was determined or could explain variation in the gut microbiome. And what we found is that host phylogeography actually explains variation that can't be explained by other factors. So altogether, all the variables we consider explained 12% of the total variation in gut community structure. And of that, 3.4%, or roughly 30% of the explained variation is coming from variation that can only be explained by host phylogeography. And that doesn't seem like very much variation, but consider that that same pattern, that east-west divide, explains only 6.6% of host genetic variation. So similar levels of variation explained in both. But this pattern is definitely reiterated. Spatial variation, the large-scale spatial variation, largely can only be explained by host phylogeography. So it's exhibiting this east-west pattern similar to host phylogeography, but it can't be explained by present-day environmental variables or differences in the environment. And more explicitly linking these changes to host demographic history, it seems we found some evidence that the microbiome diversity, or alpha diversity, reflects the historical range expansion of those hosts. Spatial patterns of host genetic diversity are the result of, or we think that they appear to be the result of historical range expansion from glacial refugia in the north. And what you get during range expansion events are successive loss of genetic diversity radiating away from these refugial populations due to genetic drift, due to founder effects compounded over longer distances. And we found that actually host genetic diversity was the best predictor of microbiome diversity, or alpha diversity, and that they exhibit that same spatial pattern to an extent. So this is only, basically it's exhibiting a spatial pattern that is explained best by host genetic diversity, irrespective of environmental conditions, which directly implicates genetic drift possibly acting on the microbiome. And that can happen in two ways, right? So either directly through the loss of these microbiome, or these host-specific bacteria that once they're gone, just like host alleles, you can't get them back because they don't exist in the environment. Or you could lose variation as you're expanding away from the refuge. Anyways, overall, it seems that the Otis jauntus gut microbiome appears to reflect host evolutionary processes. And really our study provides some of the first evidence that genetic drift can or has possibly acted on the microbiome. And then this is a mechanism of change over time. Because these patterns right here, that are this pattern, which reflects both host phylogeography and the microbiome to an extent, we largely don't need selection to explain it. That's not saying that there's no selection going on, but all you really need is genetic drift to explain this pattern, which suggests that genetic drift has created similar patterns. It's acted